All right, we are continuing, and in this video, we're going to talk about our adaptive immunity. So, our adaptive immunity contains three main components: T lymphocytes, antigen presenting cells, and B lymphocytes. And each of these have a slightly different role. Antibodies are also going to play a role here, although they are produced by our B lymphocytes. So they'll be discussed with that particular topic. Um, so like I said in a previous video, the adaptive immune response is very specific. This is where we have very targeted responses to bacteria, targeted responses to viruses, so on and so forth, as well as actual memory to these different pathogens that we come across. And this specificity makes our adaptive immunity very, very effective. So that specificity and memory makes it really effective. And so this can be really, really good for a lot of pathogens that a human is going to see repeatedly, which is part of how our adaptive immune response ended up evolving over time. And so our antibodies and our memory cells and stuff like that are a really key component of it, even though they're not directly going to be killing off cells necessarily. We still need those components of our immune response overall. So basically, in general, once a pathogen kind of gets past this innate immune response line and we get this adaptive immune response triggered, innate immunity will still continue. These cells will all keep working. They're not just going to stop once the adaptive immune response starts. So they will keep going, but then they kind of get reinforcements in essentially. And so these T cells are largely going to be the cells that are going after pathogens, actually killing them and stuff like that. Our antigen presenting cells, the name is pretty self-explanatory. They're going to take special cells from the surface of a pathogen, and these are going to be molecules that are basically like little name tags, little ID tags, and they're basically just going to hold them out to B and T cells, say, hey, this thing is here, do what you need to do with this. And so their name is pretty self-explanatory. And then our B lymphocytes are largely going to be our memory cells and producing antibodies, which will attach to those special surface proteins, those name tags, and will help further signal to a lot of these other cells, T cells and the innate immunity cells, to come and destroy that particular pathogen or infected cell. Another really important part of our adaptive immune response is the ability to tell self from non-self. All right, so this is the part of our immune system that learns to recognize which cells are our natural innate cells that are supposed to be there, and what are cells that are mutated, they're cancerous, they're infected, any debris, anything that's foreign and not normal is recognized by our adaptive immune response. And obviously, this can have some mistakes in it. And that's where we get into autoimmune disorders, which we'll talk about in another video. And we've touched on some of these a little bit in other body systems already as well, but we'll talk about them again here. But that breakdown in recognition of self versus non-self is going to lead to those autoimmune disorders, as well as some of our allergy responses. So, and that includes the innate immune system as well, is recognizing harmless things like pollen as a foreign invader that is here to do a lot of damage, when in actuality it would not do anything. It could be dealt with just as general debris that can be excreted through mucus or blocked with hair. All right. So, looking closer at some of our responses here, with these B lymphocytes especially. Actually, we're going to talk about T cells first, so I'm going to come back to this slide. So, our T cells are named T cells because they finish developing in the thymus. So they start out their development in bone marrow and then finish out in the thymus. And so they are going to be a really important part of our adaptive immune response. When they are first exposed to a particular pathogen, that's always usually going to be a much more severe disease because they don't recognize it yet. And so they are going to end up getting stored primarily in the tonsils, these naive T cells, and get activated when a specific antigen shows up and is presented by our antigen presenting cells. And then these will start very rapidly dividing through mitosis. And so those clones, those daughter cells, are going to be able to recognize that specific antigen, that specific name tag on the pathogens and stuff like that. And that becomes very hardwired into its DNA, which that whole process is called clonal expansion. So we make a bunch of clones so that we can have a ton of cells that recognize that specific name tag. We end up getting both memory T cells and T effector cells produced by this primary adaptive immune response upon first exposure to a particular pathogen. And these effector T cells, they are the ones that are going to go after that pathogen. And then the memory T cells are going to hang out for quite a bit longer. With certain pathogens, they can actually stay for the rest of our lives and then continue dividing when that pathogen maybe presents itself again. But these effector cells are going after those pathogens during that acute actual infection. And there are 3 subtypes of these T effector cells. They are helper T cells, cytotoxic T cells, and regulatory T cells. So helper T cells, as their name implies, they are going to be helpers. They are actually going to be the primary ones interacting with our antigen presenting cells. These tend to be macrophages and dendritic cells are these antigen presenters. And these helper T cells will activate these other types of T cells as well as certain B cells to start that response to a specific antigen, that specific name tag. But they can also help activate some of these innate cells as well through a lot of those signaling molecules. Cytotoxic T cells, cyto refers to cell, toxic is pretty self-explanatory. These are cells that are going to kill other cells and primarily kill infected cells of the body. And then sometimes that will be bacterial cells, which are actual cells all by their lonesome. And they're going to function very similarly to our natural killer cells. And so those are pretty similar, but very, very specific to a particular pathogen, whereas the natural killer cells just kind of go after any pathogen regardless of what it is. The more cytotoxic T cells we get, the more effective our immune response is going to be. And so they can keep destroying infected cells before they can actually have that pathogen reproduce and get out of that initial cell and into the rest of the body to infect other cells. So these are really, really important. And they're actually very, very critical to the viral response. So our response to viruses. And there is some speculation, a theory out there that the main reason that we have cytotoxic T cells is because of the viruses that have evolved with humans to infect us. And then regulatory T cells, also sometimes called suppressor T cells. These are more recently discovered, so we don't know a ton about them just yet. But they can suppress other T cell responses. And this can be helpful in some cases because during that clonal expansion earlier on, if we were to let that go on completely uncontrolled, that can lead to those autoimmune disorders, among other medical issues. There's a couple disorders that can result from the immune system being way too active during a certain infection. All right. Now we'll move on to talking about our B cells and antibodies. So, antibodies were actually one of the first parts of our adaptive immune response to be identified and characterized. And so, this came from some knowledge that individuals that survived a particular infection were generally immune to reinfection with that exact same pathogen. And so these antibodies actually function very much like the antibodies we discussed last week with blood cells during blood typing. And so they're going to function the same way. They'll recognize a particular antigen or ID tag on the surface of a pathogen. They'll attach to it, and they're going to be sticky. And so we get a lot of clumping of those pathogens, which makes it a lot easier for those phagocytic cells to come through and kind of gobble up big clumps of these different pathogens all at the same time, as well as helping to signal different other cells to come start attacking them. And so now that we know that agglutination, that clumping is happening, these antibodies are also sometimes called immunoglobulins. So globulin is that glob, that clump, but these are also proteins that get produced by our B cells. Once we've produced antibodies to a certain pathogen, they get produced by B cells, and then they just kind of float around in our bloodstream and stuff like that, in the plasma, and then we'll be able to very quickly signal the immune response if we were to get infected with that pathogen again. So B cells also undergo clonal expansion once they've been activated. And so they're going to start out as naive cells as well, just like our T cells do. So they are uneducated at that particular point in time. They don't know what they're looking at with a particular pathogen yet. But once they're taught what to look at, we're going to go ahead and make all of those clones so that we have a bunch of cells that recognize that particular pathogen again. And so the memory B cells are going to undergo a very similar process to the memory T cells, and this ultimately gives us a much stronger and faster immune response if we are to be infected with that particular pathogen again. So those memory cells, they are going to produce some of those antibodies. We make clones of those, and those clones will keep expanding, keep making copies of themselves, along with those antibodies on them that can then help float around out in the bloodstream to be basically a surveillance system at that point. On the other hand, our plasma cells, which are going to be the rest of these cell types, these are going to be producing and secreting most of the antibodies during the active infection. Once they've been activated, once they've been educated on what to be looking for, after they make a ton of antibodies for a while, they will die off because most of their energy gets devoted to making those antibodies, and so they can't really survive for much longer. But they're making tons and tons and tons of antibodies so that we have all of those available during the initial infection, as well as having plenty to keep around afterwards as part of our surveillance system. So we've got that primary response, that education stage, and then that secondary response of our clonal selection, our clonal expansion, making lots more copies of those as needed. And so B and T cells are really essential again to that adaptive, that specific immune response, because all of these memory B cells, they remember that one pathogen specifically for the rest of their lives. These antibodies are to that one specific pathogen for the rest of their lives, so on and so forth. And so If we were to get reinfected with that same pathogen again, these antibodies and the memory B cells are going to recognize that quickly, and they are going to ultimately trigger an advanced response in that innate immunity, as well as triggering a lot of those T cells, especially those effector T cells, to really get going and start taking care of any infected cells or any of those pathogens that are found free floating around in the body. All right, we will wrap up this video here and continue in the next one learning about lymphatic vessels.